Intelligent that survives. It is the one that is most adaptable to change. Charles Darwin. With this notion, I greet a very good afternoon to one and all present here, and I wholeheartedly welcome everybody to the International Virtual Conference on Biology in 21st Century, organized by the Department of Zoology of Kongunadu Arts and Science College, Coimbatore, Tamil Nadu.
Prayer is a positive vibe. The good starts always begins with the blessings of the Almighty. So I pray the Lord to shower His blessings throughout the entire session. The time has come to ravishingly welcome you all to this program. Now I kindly call upon our respected assistant professor and head in charge of the Department of Zoology, Dr. S. Bunnikumari ma'am, to deliver the welcome address. Please, ma'am.
Good afternoon to one and all join with us for the International Virtual Conference on Biology in 21st Century, organized by Department of Zoology, Kongunadu Arts and Science College, Coimbatore, Tamil Nadu, India, from 4th August to 30th August 2021. It gives me great pleasure to extend a very warm welcome to our esteemed Honorable Secretary and Director, Dr. C.A. Vasagi Madam, distinguished Principal, Dr. M. Lakshmanasamy Sir, most valued, respected our Emeritus Professor, Dr. Murugan Sir, respected Dean Research and Development, Dr. Palasamy Sir, Dean Academics, Dr. Madan Shankar Sir. I also extend my warm welcome to our guest of honor, Dr. Jose Teixeira, Research Scientist, Center for Neuroscience and Cell Biology, Center for Innovative Biomedicine and Biotechnology, University of Coimbra, Coimbra, Portugal, who is with us today to share his expertise on toxicological and pharmacological effects of new molecules. I also greatly welcome all the invited speakers of this virtual conference. And also, I wish to welcome the faculty members, research scholars, and the students of Kongunadu Arts and Science College and other academic and research institutions. I also take this opportunity to welcome and thank our organizing team for organizing such an informative webinar, which gives an opportune time to review contacts and discuss problems of mutual interest with delegates. It is gratifying to note that the agenda of the virtual conference covers a wide range of very interesting topics. I once again, on behalf of the Kongunadu Arts and Science College and on my own, I welcome you all to the International Virtual Conference on Biology in 21st Century. I wish everyone to have a great time with us today and forthcoming days. Thank you. Thank you, one and all.
Thank you, ma'am, for your warm welcoming.
A leader is the one who knows the way, goes the way, and shows the way. Now, I kindly request our beloved principal, Dr. M. Lakshmanasamy sir, to deliver the inaugural address to the gathering. Please, sir.
Thank you, Binukumari Madam. Good afternoon, one and all present here. Most respected Secretary and Syndicate Member of Bharatiya University, Dr. C.A. Vasagi Madam. Speaker today, Dr. Jose Teixeira from Portugal, and other guests, Dr. Srinath Sridharan from Singapore, and Dr. Soundararajan Nagasundara Pandian from South Korea, Dr. Gima Godan Raman from Germany, Dr. Kaushik Nayak from New Delhi, Dr. Ramachandra Nagaraju from Bengaluru, Dr. Ines Miranda Santos from Portugal, Dr. K. Murugan, former Vice Chancellor of Tiruvallur University and former Registrar of Bharatiya University, Dean Academics, Dr. S. R. Madan Shankar, Dean R&D, Dr. S. Balasamy Sir, HOD in charge, Dr. S. Binukumari, Organizing conveners Dr. P. Bharati and Dr. S. Manivannan, faculty members, research scholars, PG, UG, and other participants. On behalf of the management, welcome you all for the International Virtual Conference on Biology in 21st Century from 4th August to 13th August. Coming to the subject, why is biology important? So, as a field of science, biology helps us understand the living world and the ways its many species, including humans, function, evolve, and interact. Advances in medicine, agriculture, biotechnology, and many other areas of biology have brought improvements in the quality of life. The 19th century, the age of engineering; the 20th century, the age of chemistry and physics; the 21st century, the age of biology. Life sciences offer exciting opportunities. Example, reproducing organs using stem cells, merging natural and synthetic tissues, applying personalized medicine, repairing genetic disorders, curing neurodegenerative diseases, deploying nanotechnologies in medicine, enhancing crops with the biotechnologies, example, drought-resistant plants, producing third and fourth generation biofuels. Forum on Global Biotechnology, Paris 12. The world has become much more complex and the pace of technological change has increased. Lack of information about the benefits of the new technologies is always the risk. But many people are rather reluctant or even opposed to new technologies. So the world has become much more complex. What do we need to fully develop the opportunities offered by life sciences? Smart scientists who develop, smart politicians who regulate, smart citizens who accept. We need to communicate proactively, otherwise, others will fill the void. We need to speak a language everybody understands. We need to show empathy for public concerns and avoid being perceived as arrogant. We need to raise our voice when science is being misused or evidence being ignored. So, with this few introduction, I am also eagerly waiting for the guest speech. The time is more precious. Once again, on behalf of the Zoology Department and management, I welcome you all for this virtual conference. Thank you. I wish the program a grand success.
Thank you, sir, for your encouraging words.
Now, I kindly request former Vice Chancellor of Tiruvallur University, Vellore, and former Registered in charge of Bharatiyar University, Coimbatore, and our Honorable Emeritus Professor, Dr. K. Murugan, sir, to deliver the special address. Please, sir.
Very good morning to everybody. It is really I am very happy to participate in the virtual conference. Biology in 21st century is organized by Department of Zoology, Kongunadu Arts and Science College. It is a very leading college in Coimbatore city, textile city, as well as, and also they have the Kongunadu Arts and Science College is also having other bioscience department like biochemistry and also biotechnology, physics, chemistry, and it is a leading college in Coimbatore city, Tamil Nadu, India. And Madam, the most respected and honorable Madam Dr. Vasugi, the secretary as well as director of the institute, and she is also a biologist and zoologist. Her father also is a leading biologist and who has taken much pain to develop this college. And our honorable vice chancellor, the Bharatiyar University, he is also a biotechnologist. And madam, our honorable madam, is one of the honorable syndicate member of Bharatiyar University. The Kongunadu Arts and Science College is really progressing in the particularly in the field of biology and other biosciences. And really, I am very happy to congratulate Dr. Binu Kumari, head in charge for the conference, and Professor Dr. Lakshmana Swami. He is the principal as well as head of the department of zoology department. Dr. Palusamy sir, he's a R&D dean, and Dr. Madan Sankar, he's a dean academics, and Dr. all other staff members teaching and non-teaching department of zoology department, and also Dr. Bharati and Dr. Manivannan. They are all putting their efforts to convene this meeting. They are all conveners, and the various speakers from experts from various countries like Germany and Singapore, and in South Korea, and also the University of Coimbra, Professor Dr. Jose. He's a very good expert in toxicology and pharmacology, and the biology in 21st century. Dr. Lakshmana Swami sir rightly pointed out that the 21st century is meant for biology, and neuroscience is a multidisciplinary subject, and it belongs to anatomy and in physiology, even mathematical modeling, computer science, to know the functional aspects of the neuroscience, and also the toxicology is a very important for validation of any drug for before before putting into the market for human use. Similarly, the toxicology, pharmacology also very important, so that our Honorable Vasgi ma'am, the secretary and director of this college, has has given direction to zoology department to organize this conference. I think the deliberation and discussions will benefit to the mankind, particularly the younger generation. I congratulate each and everybody who have involved in this conference. I'm much thankful. I'm also joining this conference, so I'm much thankful to everybody. Congratulations. Thank you, one and all. Thank you very much. Thank you, sir.
Thank you, sir. Thank you for your encouragement, sir.
Thank you.
Now, I humbly call upon our beloved Dean, Research and Development, Dr. S. Palsamy sir, to felicitate the gathering. Please, sir.
Esteemed Secretary and Director Madam of our college, respected Principal, the Honorable former Vice Chancellor of Tiruvallur University and former Registrar of Bardiyar University, Dr. Murugan sir, respected Head of the Department in charge, Dr. Birin Kumari, most respected the faculty members and the organizing secretaries, Dr. Bharati and Dr. Manivannan, most distinguished guests from various countries, and dear participants, good afternoon to everybody. At the outset, it is a happy occasion for me to offer felicitation in this wonderful webinar, international webinar organized by the Zoology Department. So they have chosen a right topic to deliberate the proceeding. Actually, in 21st century is the era of biology. So 21st century beginning for the first 2 decades. Actually, the pandemic, COVID-19 pandemic, due to which almost throughout the world, the economic development due to poor development in industry, agriculture, and other areas resulted into very poor economic development. So I think for the next few
Sir,
decades,
Dr. CK, kindly mute your mic. Dr. CK, please. Kindly mute your mic.
Yes, sir.
Dr.
Yes, sir.
CK,
Oh, sir.
kindly
Yes, sir.
mute your mic, sir.
thank you, sir. Thank you. So 21st century, we have to do a lot of things for the healthcare, not only healthcare in agriculture side, in environment, in forestry, but in so many areas, the role of biology will be more. So the biological scientists, they have to do a lot of work in this area to maintain the globe in a normal way, in an equilibrium way. In this occasion, it is my duty to say something about the development of some drugs and also development of resistance against the novel coronavirus. Because nowadays, for the past 2 years, this is the problem all over the world. Scientists from different countries, they are trying to develop drugs to retaliate the virus. So usually for the development of any vaccination, it will take at least 10 years. Smallpox, it required 40 years for the standard vaccine. So, but for this COVID-19, this novel coronavirus, only within 2 years the people developed the vaccine. So, such a high technology have developed nowadays. The technology is available. So, scientists they produce this type of many types of vaccines. India is also very proud to say that we have produced one vaccine, Covaxin, indigenous vaccine. It is very powerful and fruitful against the virus. So one thing the genes from the Neanderthal people, they are associated with the human body, human chromosome, and it is giving both the susceptibility and then the protection against the virus. So Neanderthal people, they are the caveman, lived some 40,000 years back in the Neanderthal Valley of Germany, very close to Poland also. So these people, when they came to contact with the modern man, they breed themselves. And then the modern man acquired genes from the Neanderthal people, and a stretch of 50,000 genes, they of 50,000 nucleosides, they are available in the chromosome number 3 of the human being. And these 50,000 genes, they are responsible for the susceptibility of the COVID-19. But at the same time, in the chromosome 6, 9, 12, and then the 21 of human beings, they are having resistance genes. So both these genes, they are inherited from the Neanderthal people. So this push and pull effect, that will give a balance. So how in evolution the genes are selected by the chromosome in course of time. The German and Swedish scientists, they have attempted the work and they have found the genes from the Neanderthal people, they are providing resistance or protecting us. So great risk also be there by the Neanderthal genes associated in the chromosome number 3. This work has been published in the Nature Medicine in 2021. So very wonderful work. The genetic analysis have been made by the people to analyze the impact of the COVID-19 on human beings. Another one, the targeting. Targeting group is the most important because virus we can control only by vaccine or by targeting certain functional area of the virus. So all we know nowadays about the glycoprotein or the this spike protein that is responsible for the entry of virus inside the body through the ACE2. But some of the places like our stomach and intestine, they don't have ACE2 a receptor. But at the same time, the virus try to enter inside the body with the help of the nucleoproteins. That means the nucleocapsid protein or N protein. So targeting of the N protein is most essential. Many number of plants, they are having wonderful candidates to target the both spike protein as well as the nucleocapsid protein. So antiviral drugs extracted or the purified from the plants like ginger, and then the andrographis, cardiospermum, salvia rosmarinus, evolvulus alcinoides, clitoria, and then the Strobilanthus, Eugenia, Rhesidae, and then many number of species, Phoeniculus. So many number of plants we have listed in our study, in our early study, we have listed in Western Ghats alone, particularly in Nilgiris, more than 150 plants has antiviral property. So the thing is, we have to go deep into the plants for research, and our insight will be more in these plants to explore the biochemical compounds and how they can function against the virus like that. So new scope is there in these plants. You know, India is a mega diversity nation with a lot number of plants, not only the common biodiversity. Many number of plants, more than 7,000, they are medicinally important. Proper biochemical explosion is more important to use the plants for the practical purpose. So this is one area biologists can do in future regarding the preparation of the medicine against the novel coronavirus. And another important area, the diet for the dementia. You know, the post-COVID-19 problem is so severe. You know, this black fungus, nowadays it is mostly affecting the people after COVID-19. Similarly, the dementia, it is the memory loss and associated problems that is also happened. The study by the the Weizmann Institute in Israel, they have studied that more than 88% of the senior citizens in India, they have affected by the dementia problem. So for this also, dementia, how it is happened, simply the deposition of the insoluble plaques in the nerve cells, in between the nerve cells. When the plaques are heavy in density, definitely the problem will arise. So this is one thing we have to concentrate in future. The beta cytosterol. Beta cytosterol is obtained from plants. Many number of plants in the form of phytosterols they are having. So phytosterols are the only remedies for this type of the dementia and also the Alzheimer's disease and other memory loss problems. So many number of plants, more than 100 plants are available nearby our Western Ghats ecosystem. Definitely exploration, the biochemical exploration and further studies, clinical trials, they will be more useful. So 21st century beginning, we have to concentrate in this area. And the drug, now, even now, some of the plants, they are yielding some drugs like the niclosamide. Niclosamide is mainly used for the tapeworm problems. But recently it has been known this is also useful to control the novel coronavirus, because this novel coronavirus in stomach and then the intestinal area, when it tries to enter inside the host cell, there is no ACE2. But it tries to enter inside the body at the time. These viruses have been engulfed by a vesicle which is acidic in pH. But this niclosamide, they can alter the pH and arrest the entry of the virus. So very normal. The drug that is now commonly used by the people, that can also be used to arrest the entry of the virus. So many such things, even when you go to agriculture side, so due to breeding and other technologies, people developed a lot of new varieties with a number of desired characters. One such variety developed by the Center of Cellular and Molecular Biology, Hyderabad, the Samba Masuri. Samba Masuri is developed from the traditional varieties of rice. It has the genes responsible for low carbohydrate, low content of carbohydrate. The plant is drought resistant also. Paddy is normally the water requiring plant. But this variety developed by the center is a drought resistant plant, as well as that can have low carbohydrate. So this is most useful for the diabetic patients. So these kinds of researchers, all over the world, India is the capital for the diabetics. So this kind of research to promote the plants like the Samba Masuri and other traditional varieties with low carbohydrate, with high protein, that can be used in future. So proper research in this area are most important. In an agriculture side also, the conservation of water, that can also be done by physical engineers. But anyhow, it is also associated with biology. So numerous things are there in the area of biology, even in environment also. For environmental problems also, only thing, increasing the green cover. So to enhance the carbon sink, we have to raise the forest cover further 2.5 to 3.5 million hectares in India. Only by planting suitable tree species, air pollution tolerant tree species, we can tackle the problem. So many research are needed in biological science in 21st century for the comfortable life of man and other living systems. So the Zoology department organized a wonderful e-seminar or e-conference, biology in 21st century, a most suited, most required topic. The young researchers, definitely they should know about the importance of biology, how it is playing role in human life, and what kind of things required from the biological sciences, not only for becoming entrepreneur, but also for the welfare of the human being. In biology, many areas are there. The healthcare, environment, forestry, human health, so many areas are there. So proper research is required. So definitely the young people will get benefit, because the guest speakers are from various countries and they are very expert in their area. Definitely this will be more useful conference for the participants. I hope the participants will interact with the resource person to acquire knowledge and to get technology developed there. So definitely it will be useful for your career. So I congratulate the entire team for organizing this wonderful conference. I wish the program a grand success. Thank you for the opportunity. Thank you. Thank you very much.
Thank you, sir, for your motivating speech. Now, I'll call upon Dr. P. Bharati, Assistant Professor, Department of Zoology, and the convener of this conference, to deliver the dynamics of the conference and also to introduce and welcome our today's honorable resource person, Dr. Jose Teixeira, Research Scientist, CNC, Center for Neuroscience and Cell Biology, CIBB, Center for Innovative Biomedicine and Biotechnology, University of Coimbra, Coimbra, Portugal. Please, ma'am.
Good afternoon to one and all present. Join with us for this virtual conference on biology in 21st century, organized by Department of Zoology, Kongunadu Arts and Science College, Coimbatore. Honorable Secretary and Director, Dr. C.A. Vasigi Madam, most respected Principal, Dr. M. Lakshmanaswamy Sir, and Honorable our Emeritus Professor, Dr. Murugan Sir, and respected Dr. Palswamy Sir, Dean, Research and Development, and Dr. Madan Shankar Sir, Dean Academics, and most valued guest of honor, Dr. Teixeira, and participants. It's great honor for me to propose the dynamics of the conference as a convener of this conference. The conference will be held from the 4th to the 13th August 2021. We have seven eminent speakers from various countries around the globe, and we have received 60 abstracts from various scientists, faculty, and researchers, and the students from various institutions for the oral presentation. And I'm very glad to say that we will be having oral presentation during this conference as well. And it's time for me to introduce our today's speaker, Jose Teixeira. He is currently working in the University of Coimbra as a researcher. So he got graduated from the University of Porto, and also he finished his PhD studies in pharmaceutical science in the year 2016 from the same university, that is University of Porto. And after that, he carried out several national and international scientific internship in various places such as Cambridge, United Kingdom, San Diego, Spain, Coimbra, Portugal, and the Netherlands to obtain the advanced training in medicinal chemistry and mitochondrial biology field. And along with that, he is also worked as a postdoctoral researcher at the Faculty of Science at the University of Porto during the year 2017 to 19, and also at the Center for Neuroscience and Cell Biology at the University of Coimbra in the development and the biological evaluation of new chemical entities targeting the mitochondria in a search for a potential therapeutic solution for pathologies related to oxidative stress. And to his credit, he has 29 peer-reviewed publications and 3 book chapters and 47 oral presentations. Among these 47 oral presentations, he has been invited to give the presentation for 4. And then he is also having the 55 poster presentation in various scientific meetings during the year 2010 to 2021. And also he filed 2 patents from his work, and he is having the H index of 15 in the Web of Science. Currently, he is a postdoctoral researcher at the Center for Neuroscience and Cell Biology at the Coimbra, where I have done my postdoctoral as well. JT research interest is focused on unraveling the underlying mechanisms behind the pathophysiology of non-alcoholic fatty liver diseases. So with this brief introduction, I hand over the session to Dr. Jose. Please, Jose, you can carry on with your presentation.
Thank you. First of all, I would like to kindly appreciate the kindly invitation and the introduction. I congratulate all of you for this organization, you for contact me, and the university for this huge international meeting. I think it's a very important. I think it's very important to have these meetings, especially for young students, to get more insights on biology. So are you listening me okay? Are you seeing my presentation?
Yes, Jose, we could see. It is going well. Thank you.
Yeah.
Yeah, it's going well. You can carry on. Yeah.
Okay. Yeah. So I will talk about my work and the work I've been developed for the last years, especially on toxicology and pharmacology of new molecules. So briefly, I will talk about toxicology, pharmacology, drug discover and a drug development process, the disease I'm interested for in my research, non-alcoholic fatty liver disease, the role of this subcellular organelle called mitochondria in non-alcoholic fatty liver disease, and then trying to find new solutions for this disease, bridging the gap between nature and the antioxidants. So I heard this morning that biology is very important field. I totally agree, but I would like to have also medicinal chemistry in this topic. I will not go into much detail on this, but for example, medicinal chemistry is a multidisciplinary field of chemistry that involves biology, science, pharmaceutical science, and medical science. So it concerns with invention, discovery, design, identification, and preparation of biological active compounds and study their metabolisms. So I think it's also very important to have this in mind, and also important to add at this conference in biology, because it's a multidisciplinary, and one of the main aspects that I would like to address is the multidisciplinarity of our work. It's I think it's our work of the work. It's more valuable than you if you work alone. So I heard this morning a sentences of Charles Darwin, but I also like to when I talk about toxicology and pharmacology, I also like to have this. To mention this gentleman, this Paracelso, that was the father of all modern toxicology, that says that all things are poisoned and nothing is without poisoning. Only the dose makes a thing, not a poison. So this is a very important concept in toxicology and pharmacology, because this is the main center of everything. We know now that obviously there is also other factors that affects toxicology and pharmacology, such as time and time of exposure. So in terms of general aspects of toxicology and pharmacology, we can have, for example, an acute exposure and an acute effect. We can have chronic exposure, so exposure that persists on time, and then we can have chronic effects of toxic or pharmacological drugs. We can also have chronic effects, but acute chronic exposures, but then acute effects on time. And then this is the most interesting case on pharmacology, that we have the case when we have acute exposure, but then the effects are acute indeed, but just delayed on time. This is the more difficult aspects of toxicology to deal. I just bring these examples, especially for new researchers or for young people, that reinforce the idea of Paracelsus. So everything is poison. Only the doses make difference. So I would not recommend you, for example, to drink 6 liters of water in the once. So, or 118 18 coffees, or 13 alcohol shots. So the consequences for your life will be really dramatic. So please do not do this at home. But this is a very interesting, so very familiar substance that we normally consume, are very toxic depending on the doses, obviously. But then obviously we have also, I bring also other examples, more toxic than water. Obviously, for example, we have sorry, we have here cyanide, which is very toxic drug used in the Second World War. We have nicotine from tobacco. We have sodium chloride, obviously, in a huge amount compared with cyanide. Sodium chloride that we used normally to cook our food. And then, for example, sucrose, sugar. In a huge amount, it's lethal, but it's lethal as well. So sugar, please take this in consideration because it will be important for in the future of my presentation. So everything is just to restrain that everything is toxic depends on the doses. So my advice is take a moderate life without excesses. So, but in fact, what is a drug? So a drug is a substance that is primarily used to bring about a change in some existing process or state, be it psychological, physiological, or biochemical. So we can divide these in three main parts. Let's say that. So there is a chemical structure. Okay, the chemical structure of a drug can contain alcohol, ether, and amide, carboxylic acid in the structure that will be important for the therapeutic action or the molecular mechanism of action. So you have the chemical structure, then we have the therapeutic application for this drug. It can be a spasmolytic, it can be a antidepressing agent, myorelaxant agent. So there is a cardioprotector, beta-adrenergic compound. So the therapeutic of action depends obviously on the chemical structure. But also the molecular mechanism of action also it's important. So the drug, is it an enzyme inhibitor? Is it a protein activator? Does it change the membrane receptors? Okay, so these three components, when we try to develop a drug or find a new drug, it's these three main things, among others, obviously, three main things that we should have in consideration. Presenting like this, it looked like simple to find a drug or to have a drug for a specific target or disease. But in fact, this is not very easy. It's very complicated. I also like to present this. This is a map of London Metro. Well, it's not too complicated, but when you go there and you start to try to find where you want to go, it's really complicated when you go to the first time. It's very complicated. So if you are not aware, you easily miss your stop and you are lost in the middle of this labyrinth. So drug discovery and development, it's pretty much like that. So it's very complex. There is very high ways. Sometimes there's traffic, so it's complicated. And then in most of the times, potential molecules lost in the middle of the process. For example, this is an example that I also, and I think is what it was already mentioned, related with vaccines in the introduction of this seminar. Is that, for example, developing a drug or a vaccine or a molecule, it takes at least 10 years. What the scientists did with the COVID vaccines were pretty amazing. But in general, it takes at least 10 to 12 years to have a drug or a molecule approved in FDA agency to be administered in a disease. So, but to have an idea, for example, to have one molecule approved, normally we started with 10,000 compounds in the drug discovery process. That then it works like a funnel, a tunnel. So in the first stage, we have, for example, more than 10,000 compounds that are sequentially being lost because they have no efficacy, low bioavailability, etc. So then the preclinical stages, we have just dozens of compounds, and clinical trials, we have just a couple of compounds that involves the volunteers, phase 1, the clinical trials, phase 2, efficacy, small scale, and phase 3, efficacy on the large scale. Then we have the submitted paperwork in FDA or other agency, the review that could take 2 or 3 years, and then the large scale manufacturing phase production that is also it could take also 2 years. This is what also once again, it was wonderful that we did. There is countries that in which the vaccine of COVID is not being well distributed, but at least in Europe and US, it's been a huge work being done. Okay, so this is a long process. It took, say, too much time, too much work, and obviously it's a huge amount of money that is involved. And obviously, the big pharma at the end wants to revenue the investment of this process, trying to increase the amounts of the price for the drugs. This is so the less expensive phase is the drug discovery phase, and it's also very crucial. It should be taking something in consideration to decrease the risk of insuccess in this clinical phase 1, 2, and 3. Despite that, as I mentioned before, most of the compounds that are developed does not become a medicine. Okay, and this is a fact. The people, I think it was already mentioned this morning as well, that for example, disease like dementia has few therapeutic options. There is several diseases that with a few or nothing, or non-therapeutic solutions. So this is critical for the well-being for the well-being of the people, and for the healthy care systems. So why do these compounds do not become medicines? There is several reasons. Sometimes we have complex disease targets. For example, Alzheimer's, Parkinson's disease is very complex. Sometimes we have the costs. Sometimes we have adverse reactions of the molecules in clinical phases. We have poor absorptions. We have molecules that are not effective enough. Just they do not stop the disease. Just delay or avoid symptoms. Not selective. They have huge side effects. They are not safe. They are not stable. And this topic I bring here, it's not the main topic of my talk. I bring this topic here. The intellectual property is very important. It's very important because obviously big pharma wants to have revenue on the investment. So if the intellectual property of the molecule that we find or we develop is not protected, so there is no value for big pharma, because they will not have some revenues on this, because anyone can produce the molecule. So this is one of the reasons that the molecules also do not become medicine. So, and sometimes the scientific community or the medical community has to put it in a balance what is desirable. So sometimes, so I bring here the black box warning for hepatotoxicity and the black box warning for cardiovascular toxicity. These are approved drugs, previously approved drugs for different clinical conditions, and in red I present those who were retreated from the market, or they are still in the market, but they present cardiovascular or hepatotoxicity. So in this case, we have to see what happens, which is more benefic, the risks or if you want to take the risk or not. And it happens the same, for example, with the coronavirus vaccines. It's obviously that we don't know yet the long-term effects of these vaccines. I believe that they are pretty safe, but we don't know the effects of long-term effects of these vaccines. But the benefits were higher, definitely higher than the costs. So we have to put these vaccines on the market. The big pharma had to put these vaccines on the market to try to control this pandemic crisis. So I hope that the coronavirus vaccines will not bring to this list. But sometimes there is a risk that has to be taken because the benefits are higher than the costs. So we can somehow control these or try to control these to avoid some of these effects on this process, on this drug discovery process. Okay, so this is I can divide the other graph in 3 phases. So it's exploratory discovery, when we look for the disease, the targets that are identified in disease, and then we have the drug discovery phase. So this phase could take a couple of years, but it's cheaper than the later phase, because we just have a couple of grams of molecules to try to see the efficacy, toxicity, pharmacology effects, side effects. So this can be seen as a cycle. So we have a molecule, we perform safety studies, demonstrate the effectiveness of this. If it's not good, we can go back and try to chemically modify the molecule, try and improve safety, try and improve effectiveness. And then if it's not good enough, so we can turn this in a cycle and could extend a couple of years. But with this, if we have a rational drug design and a drug discovery process, we are more close to the success, or if you want to say, we can decrease the risks of failure in clinical trials or in registration in FDA or European Agency for Medicines. So something that we should have in consideration during this process. So this is as I mentioned before, there's a lead optimization process for the compounds, the drug candidate confirmation with effectiveness and no toxic effects, and then the preclinical and clinical characterization of these molecules before the submission for approval. So something that we should have in consideration: efficacy assessments. Does molecule work? Is effective or not? Absorption, distribution, metabolism, and excretion profiling. How can this molecule be delivered, and what does to the body? Okay, and then obviously toxic and pharmacologic assessment. Is this safe? Is the benefit clearly higher than the costs? Okay, this is something that we should think about it before we start the process. And then is the manufacture viable and controllable? Can we scale up to tons of this molecule? Well, this is something that we should have also in consideration. So and then it comes this pharmacodynamics, the mechanism of action of the molecules in the body, and the pharmacokinetics in the pharmacokinetics concepts in absorption, distribution in the body, metabolism, and excretion. So let's think about it on drug discovery and development. So to be more efficient and try to reach the target. So first, what is your target? So What is your target? Is a protein? Is a organelle? Is a cell? A cell, a neuronal cell, a hepatic cells, cardiac cells? What is your target? So, in taking consideration once again these patentability issues, where is your target? We have to think about drugability. So, is in the liver? Is in a heart? Is in the brain? Is in the skin? So we have to have this in consideration because, for example, for drugs for the central nervous systems, we have to have a huge consideration the bioavailability and that these molecules have to cross blood-brain barrier. So permeability, bioavailability, and metabolic stability. If it's really, really important, the stability. And obviously, how your drug will be used. So is it injectable? Is it a solid molecule? For example, we should have this in consideration because if you want to have a molecule that will be orally or taken by oral via, so it should resist, for example, to the acidic pH in the stomach. Otherwise, it doesn't make any sense. If the molecule will be an injectable, so in theory you don't have this. In theory and practically, you don't have this problem. It goes into circulation, and this problem will disappear. So, three main topics that should be in consideration: what is your target, where is your target, and how you think the drug will be used in the future. If you think this in advance, you are one step closer for the success. So, in a general scheme, the workflow works like that. We have a compound, find, for example, it was already mentioned in the opening session. So if you have discover a lead compound from a plant, from marine algae, etc. So if you have the lead compound, so you have to study the molecular and verify physicochemical properties of these molecules, and then try and then do the biological screening against the target you aim for. Okay, if it's good enough, you can move forward, but probably if the molecule is fine, it's fine in nature, is not good enough for human beings. But okay, so you do the biological screen against your target and side effects target, and then we start to optimize this molecule in a vicious cycle. So then you screen again if it's okay with that. So just move from molecular modeling, improving selectivity and potency. Just as soon as you have find the pharmacophore molecule, you have to perform tailor chemical adjustments in the molecule to increase potency, selectivity, and decrease side effects. So basically, when we have drug design, so the aim is optimizing binding interaction with the target. The reasons why we want to do this, it's we have to increase the activity in reducing dose levels to avoid side effects, and we have higher selectivity. Okay. In terms of chemistry, I will not go into detail. In terms of chemistry, we can do a lot of things a lot of chemical modifications to have this having this in mind. Okay, so drug discovery process is indeed the search for a needle in a haystack. Okay, so sometimes it's complicated, you're lost in the middle, and this is the case of this disease, non-alcoholic fatty liver disease. And I will give you some facts on this disease before I start to briefly describe disease. So let's to get some facts. There is no FDA-approved drug therapy for non-alcoholic fatty liver diseases. So there is no proven preventive drugs for these diseases. So we have a problem here. And that is pressing for finding new preventive disease-modifying drugs. But let me explain you briefly what is non-alcoholic fatty liver disease. This disease is normally associated with bad lifestyle habits, sedentarism, high intakes of calories, sugar, fat, So I will not publish any specific drink, but drink with high amounts of sugar, chocolates with high amounts of sugar, burgers with high amounts of fat, sedentary life, no sports. So it is have been associated with non-alcoholic fatty liver disease. This disease is a progressive disease. So it starts with accumulation of fat in human hepatocytes. It starts with simple steatosis, then progress for steatosis plus inflammation, then steatosis plus hepatocyte injury, that's ballooning, then moves to steatosis plus fibrosis that can progress to cirrhosis, hepatocarcinoma, and finally death. So the scientific community says that until this phase 2, 3, this disease can be reversed. As long as we have fibrosis, there is no way back. Okay, we can stop this molecule. We can stop the progression of the disease until this phase. As long as we get fibrosis, and then there is no way back to stop this to going to cirrhosis or hepatocarcinoma. Okay, so our therapeutic window is something around this phase 1, 2, and 3. Okay. In terms of prevalence and just to see how this is problematic. So in terms of prevalence, there is 1.8 billion people with the diagnosis with this disease. Although the diagnostic is a lot of the diagnosis is not consensual in all the regions of the world. So these values can be effectively higher than 1.8 billion people, which is a prevalence rate of 25%. So just to see, for example, the most dramatic case in South America, 30.5%, 45% average, and the Middle East, 31.8% in average. So this is the leading cause. This disease is the leading cause of chronic liver disease in children, adults in Western world, and the third most common reason for liver transplants in the United States. So obviously, not all of these 1 billion people have death by non-alcoholic fatty liver disease. So in terms of progressions, the numbers that are registered are, so 25% have this disease. From this, around 60% progress to NASH, non-alcoholic steatohepatitis. From this, 41% develops into fibrosis. That from this, 40% becomes cirrhosis, which increase the risks for liver transplant, cardiovascular disease, and infection mortality. So the numbers are not so high on this, but still they are very high numbers on people suffering from this. And obviously there is also cardiovascular effects associated with which increase the risks of death. This is very problematic, and something should be done on this. So the pathophysiology of this disease is not consensual in the scientific community, but free fatty acids play an essential role on this. So this is a hepatocyte, a human hepatocyte. So the free fatty acid in circulation goes into hepatocyte, and two things can be done. So it can suffer oxidation in the mitochondrial, beta oxidation, mitochondrial peroxisomal organelles, or it can be produced triglycerides, VLDL, and export to circulation. Okay, but then this in circulation can be reuptaken by hepatocytes and then form these lipid droplets. Okay, these lipid droplets until a certain point became toxic and form lipotoxic lipid derivatives that then induce oxidative stress, endoplasmatic reticulum stress, and activation of inflammation, that goes into apoptosis of hepatocytes, release of cytokines, in general inflammation, activation of hepatic stellate cells, and then move to fibrosis, cirrhosis, and hepatocarcinoma. This is not very consensual in the scientific community, but it seems that this is the two-hit hypothesis is the most accepted one for explaining this disease. So this is another scheme of the pathophysiology of this disease. So hepatocytes have been suffer from metabolic stress due to the increase of lipid being getting into the hepatocyte, they suffer from balloon, and then we have lipotoxicity that can induce cell death of these hepatocytes. Okay. So more facts about NFLD, about drugs trying to be used in NFLD. Anti-obesity drugs, such as orlistat, no effective. Cannabinoid antagonist, such as rimonabant, alarming rate of adverse side effects on mood due to its concentration in the brain. Insulin sensitizing drugs, such as metformin and glitazones, the long-term effects, safety and efficacy in patients with NASH have not been established. Lipid-lowering drugs. So there is a concern in patients with coronary artery disease, which is a associated factor with non-alcoholic fatty liver disease. And the antioxidants, such as vitamin A, that is recommended as a first line of treatment of these patients. Confirmed there is a concern with the long-term use of vitamin A and increase in mortality detected by a recent meta-analysis. So we have problems here to restrain my first idea. We have problems here. We have no drugs. We have a huge amount of population, worldwide population, being affected by these drugs, and we need to do something, because diet and exercise are considered the first line of treatment for patients with NFLD, but their results on therapeutic efficacy are often contrasting. So diet for these people, exercise, sometimes it's complicated because it's also associated with gain weight. So it's not easy to put these people doing exercise. So despite the number of clinical trials in NFLD therapy, there is no, the therapy remains a challenge, okay? So we embrace that challenge in Portugal, in Coimbra. We embrace that challenge, trying to discover and develop new molecules that can be used in non-alcoholic fatty liver disease. So in general, we have two different approaches. One approach that is the chemistry-driven drug discovery. Well, as the cartoon shows, sometimes not the very adequate. Sometimes the big pharma use this as a strategy to revenue some money. For example, if a big pharmacy have a library of compounds that was used in the past for example, just an example, for Alzheimer's disease, but were not effective. So in trying to reuse these molecules, this library will be tested, can be tested, for example, for cardiovascular disease or liver disease. So this is a approach that is also valid. I'm not saying that's not valid, but it's something like that. So if you have developed a library of compounds to a certain disease that you showed in the past that were not effective, you can reuse these molecules to try to find best solutions in different diseases. Okay, and the biology driven drug discovery. So I think this is more rational drug design. So we have the disease selected. You try to find the target. You have to find the lead compound, and then you have the candidate, you can test your candidate. So in our case, we have the disease, non-alcoholic fatty liver disease, and we select the target. The target is the mitochondria. Mitochondria is a subcellular organelle. So this is how the books in biochemistry represent mitochondria as a bean, which is, in fact, it's not well represented, but okay. It's not a bean. So in terms of cellular context, it's more like a network. Here labeled in green, and here labeled in red. Is a network of organelles that connect each other to function well. So this molecule, so these organelles are responsible for producing ATP, producing heat, regulate redox state of the cells, regulate calcium homeostasis, and regulate cell death and cell life process. And obviously is involved in several diseases like cancer, diabetes, Alzheimer, and Parkinson's disease. So this is not a unique target for non-alcoholic fatty liver, but there is a valid target for non-alcoholic fatty liver disease. And mitochondria can suffer from oxidative damage, okay? So like happens in food, oxidative stress oxidize the molecular part of the cells that can lead to damage and eventually cell death, okay? And what is the role of mitochondria in non-alcoholic fatty liver disease? It can be seen as a Janus phase. So in the first phase of the disease, these bad lifestyles increase the number of lipids being oxidized by mitochondria. To deal with this lipid intake, mitochondria increase oxidation of these lipids and also try to increase the network to be able to read better with this increase of lipids that are reaching hepatocytes. But then somehow it's not really explained, and somehow the mitochondria cannot deal anymore, at certain point cannot deal anymore with this amount of lipids that are reaching hepatocyte, and became dysfunctional. So becoming dysfunctional, so is not effective anymore in producing ATP, and start to burst the reactive oxygen species production, and then leads to NFLD, NASH, cirrhosis progression. So this is how mitochondrial damage contributes to NFLD progression. So in fact, mitochondria is an attractive target for pharmacotherapy. So it controls several biological process. But for drug discovery, it's a very tricky landscape. Why? Because if you want, for example, to control reactive oxygen species production, so we have to have in consideration that reactive oxygen species also play a role in signaling pathways in normal cells. So we have to find a way to control mitochondrial damage without disturbing mitochondrial function. This is the tricky thing on controlling mitochondrial function. So we started with a dream that was join nature, drug discovery, and mitochondria in non-alcoholic fatty liver disease. So as was mentioned this morning, and I restrained this, nature is a huge source of inspiration for molecules, and we try to be inspired our drug development process to develop mitochondriotropic antioxidants. Okay. So we start with a lead compound that is a phenolic acid, is a polyphenolic acid or a polyphenol from the family of polyphenols. So these molecules are known to be good on free radical scavenger, inhibit lipid peroxidation, modulate gene expression, and chelate transition metals like iron. So we start on this, and then we went to the lab, trying to perform some chemical modifications, and then came up a couple of compounds that we call it mitodiates. So this is a strategic synthetic strategy for these molecules. I will not go into detail because it's not the topic of this talk, but a couple of compounds came up, and then we performed several in vitro and in vivo tests to see efficacy, toxicity of these molecules for non-alcoholic fatty liver disease. And now, in the next 10 minutes, I will show you some data from our experiments with these molecules to trying to convince you that we may have something on this to be using on this disease. So first of all, we started with cell-free in vitro tests. So trying to evaluate the antioxidant efficacy against 3 different radicals. And I call your attention for these 2 molecules, SYN6, SYN4, that has remarkable antioxidant activity against these 3 radical. These data are from free radical scavenger activity of these molecules. So then we move and we evaluated the effects on our target, mitochondria. So this is an experiment. This was an experiment performed to evaluate the amount of compound that is accumulated within mitochondria. So this is a curve calibration with the molecule, and then we energize mitochondria with a substrate, and then this is the amount that can be extrapolated from calibration curve. This is the amount of compound that is accumulated within mitochondria. And doing the calculations, it's around 2,000 times more concentration within mitochondria than extracellular mitochondria. So then we evaluate chelation properties. So all of them chelate iron. They inhibit lipid peroxidation in mitochondria membranes. And then the molecules prevent the opening of mitochondrial permeability transition pore. This is a phenomenon that is normally associated with cell death, higher oxidative damage, and cell death. So we have checked all the characteristics in our target mitochondria. Then we moved to cells, and we started to use human hepatoma-derived cells. So this is the dose response curves commonly used in toxicology and pharmacology. We see that increasing the doses of this molecule in blue, it's much more toxic than the molecule in red. So for that reason, and for obvious reasons, we select the compound in red for further studies. And what we observe, it's very interesting. So this compound, this molecule activates the NRF2. NRF2 signaling pathway is the pathway involved in oxidative stress response. So being this molecule activated, the compound then increase the number of mitochondria per cell, but also increase the antioxidant defense system of human hepatic cells by increasing, for example, superoxide dismutase activity, and by increasing glutathione levels of human hepatic cells. So meaning that these cells, in theory, are more prone to be, are more resistant to oxidative stress. And then we proved that. So then we had a mix of free fatty acid to these cells that leads to a huge lipid accumulation, and the compound decrease the lipid that are accumulated within the cells. So why this compound decreases the lipids, the accumulation in the cell? So it decreases the lipid droplets in the cell, it decreases the lipid droplet area in the cell, and also decreases the lipid droplet size within the cell. So, and we show, I not show here, but we have proved that this molecule increase the beta oxidation process in the cell that may explain these results. So then we moved for an in vivo study. I will try to put some video. In vivo studies, we show that our idea was to, so we had these normal mice. So we gave this compound orally to these mice in edible gelatin that they are very they very like to eat them. So for 2 weeks before we started the obesogenic diet, okay, the high fat, high sugar diet, and then the compound and the diets were maintained for more 16 weeks. So as you can see, they enjoy a lot to hit this molecule, or the gelatin containing the molecule, obviously. Okay. So some interesting data we found. So the obesogenic diet obviously increase a lot the body weight of the animals, but in the presence of a compound, this increase is not so high when compared with the Western diet. And as well, the same happens with the liver. So there is a huge increase on the liver weight with the Western diet that is not so increased in the presence of molecule. Interestingly, we also observed a decrease in AST and ALT markers, plasmatic markers. These markers are markers of hepatic damage. So they are decreased in the presence of compound in rats fed with a high sugar, high fat diet. So you can see here from some pictures on animals, you can see here that the liver is not as good as control liver, obviously, but it's not as bad as Western diet fed mice. In terms of histology, you can see that we quantify these images, and there is also a decrease on lipid accumulation in the presence of compound. So why we think that this happen? So first, the compound increases or improves the mitochondrial function by decreasing the consumption of oxygen associated with the oxidation process, but also increase the proteins that are involved in fatty acid oxidation, mitochondrial fatty acid oxidation and peroxisomal fatty acid oxidation. So in fact, there is a proof that also in vivo, this increases the fatty acid oxidation process to deal better with a huge increase of fats that are being delivered into the liver. At the end, also that we also observed in human hepatic cells. So this molecule increase liver total antioxidant capacity, the glutathione levels in the liver, the most antioxidant present in the hepatic cells, and the mitochondrial superoxidase activity. So once again, we believe that these mice, in this case, are more resistant to oxidative damage than the non-treated ones. So, take-home message for young people and for all of you. We believe that the rational drug discovery is crucial to decrease the failure during drug development process. Early evaluation of toxicological and pharmacological properties of new molecules might increase success in preclinical and clinical phases. And the search for druggable targets triggers innovative R&D process. In the case of NFLD, is a multifactorial condition associated with deficient lifestyles. A lot of research have been doing in this disease in different areas. We are focused on novel interventions, and we believe that mitochondrial tropics antioxidants based on dietary scaffolds potential application can be extended to the liver disease context, such as NFLD, NASH, and NFLD. So also a cartoon that is a very good message because when it comes to drug discovery, when we are not trying to make complicated molecules, but molecules that will be effective for disease and targets that we selected. And finally, I would like to acknowledge my two supervisors in Portugal from the University of Coimbra, Paulo Oliveira, and the University of Porto, Fernanda Borges, and my international co-worker, Dr. Marius Wiekowski, and obviously the group where I'm inserted to, and funding agencies for funding. And thank you for your attention.
Thank you very much, sir, for sharing your expertise about toxicological and pharmacological effects of new molecules with all of us.
You're welcome.
Now, the session is now for discussion. Participants are requested to post their questions and queries either in the chat audio to clarify your doubts.
I'm trying to read the questions. Okay, so the first question from Professor Barati. So in general, especially for liver disease, in terms of how many animal species should be tested, as far as I know, mice model is enough. Mice model is enough for this, but for example, there is some studies in that study the effects of diet and obesity in primates, but it's not essential for a drug being approved. And moreover, the fact that there is no approved drugs for FDA, for example, approved drugs for NFLD, speed up. There is a way to speed up the approved process. Okay, hopefully I will.
There's another question from Dr. Manivanan, sir.
What is this question?
What is the sequence of activities in the new drug development?
I'm not understanding properly the question, but okay. Well, I know. I will try to reply. I know that. For example, I know that these molecules are effective free radical scavengers. Okay, that's for sure. Because they have we have tested that. We have tested that on at several levels. We also think so. I call this as a multi-operational antioxidant. So as a first mode of action, so the compound is a free radical scavenger acting on its reduced form. Once is oxidated to reduce different species, it can be stabilized by resonance due to the chemical structure, and then activate other cellular mechanisms within the cell, such as NRF2, that then leads to activation of mitochondrial biogenesis and activation of antioxidant endogenous defense system. So sequentially, I think I think that's what happened. I don't know if it's a question or not from Professor, but hopefully I will reply. I replied to.
Thank you, sir. There's a question from Dr. Binu Kubari, and the question is, What are the biggest issues facing drug discovery today?
Okay, so one of them is patentability issues. Okay, so for example, and patentability issues and the risk of failure that are very high. At least in Europe and America, big pharma shut down most of the R&D departments. Why is that? Because they spend a lot of money, and they spend a lot of money, millions of dollars, trying to investigate new molecules, but then most of them fail because they try to use this chemistry driven approach. So now they try to are more connected with academia and universities to take advantage of the knowledge, high quality knowledge in the universities on drug discovery, and try to find new molecules to translate to industry. The problem is obviously, at least in academia, most of the time we are not thinking to make money, unfortunately, and we are just thinking on science and do science. For example, if we have a new molecule, but if we publish a paper with these molecules, the big pharma don't care anymore this molecule because it's published already. So there is no value to patent because it's public for everyone. And then the potential interest on the pharmacy is gone. So this is the patentability issues. It's one problem. The risk of failure is another problem. And then, for example, other big problem, it's sometimes the disease that we are looking for new therapeutic solutions are a dark, are a black hole. With this, I'm saying that, for example, for liver disease, I present for this non-qualified liver disease, I present these two hit hypotheses. That is the most consensual hypothesis, but there is a lot of controversy on this. So in fact, the pathophysiology of the disease is not clearly understood. So it's a multifactorial disease, but the mechanism, the pathophysiology is not clearly understood. So we are trying to find molecules for a disease that we know we don't understand well. So the same happens with for example, neurodegenerative diseases such as Alzheimer's and Parkinson's. We don't know the exact mechanisms, what triggers the cell death, what triggers the neurodegeneration. We still don't know this. So we are trying to fix something that we don't know what happens. So, and then this increases the risks of failure because of this.
Thank you, sir. There is a question from Kalpana, and it is there any role of genetics in etiology of NADFD, such as polymorphism?
Yes, for sure. There is a genetic component of this disease. Obviously, I not bring this. I did not bring this on my talk, but there is a genetic factors involved on this disease. It is true that it's mainly due to bad lifestyle habits, but there is a component, and the polymorphisms are important. And there is some some genes, PN3, PNP, PL3, I think is the correct one, that is associated with this disease. And the prognostics of this disease, or the speed that this goes to cirrhosis or cirrhosis or hepatocarcinoma, it's really high on these genetic factors. So, but still, and there is something that we cannot deal with. Okay, so the genetic factor of the disease tends to speed up the progression of this disease, but still a few things about genetics are well known right now.
Thank you, sir. A question from Dr. Bhupesh, and it is in silico toxicological predictions of compounds are really work of in vivo experiments?
Okay, this is a good question. This is a good question. I will say yes and no. Um, okay, okay. So in silico is in fact a very good, it's a very good, it's a very good. I'm not finding the word, but it's a very good approach to predict compounds efficacy on this target. So, but the problem with in silico, well, it's not a problem, but okay. The point is that we always need to validate molecules in vivo and in vitro experiments. Because, for example, in silico, we just have two, well, in silico, we just have two main factors, that is the molecule and the target. But in vivo, or even in a cell, in vitro, in cell culture, we have a lot of process being occurring. So the problem, my point of view, the main problem with the in silico studies is that we may lost some molecules we may lost some molecules because they are not very effective on in silico experiments. We may discard them, but which does not mean this does not mean that they will not work in vivo. I hope it's clear. So in silico is important, but to find lead compounds, I think it's my opinion. I think it's the best. Is one of the best strategies to find lead compounds. To find the best compound, I think is not the best solution, because I mean, like I said, in the cell system, in an in vivo system, there is a lot of things going on, and sometimes the biological response to a drug is completely different from what we expected in an in silico model. Okay, so I think it's a good strategy, but sometimes we can discard some molecules because they do not have affinity for a target or etc. Which means that sometimes we can discard good molecules. So it's a good option, but not definitely in my opinion, the only option.
Thank you, sir.
Okay, we have one more
Satvika
question.
has a question. Does in silico studies a better way to study about drugs and their activities?
Yeah, I think I kind of reply this question. I just have one more thing to add on this. Is that, for example, in silico are very good when we have a specific protein, for example. Okay, so so if you want to study the interaction as well as computational modeling, when we have this drug protein interaction, I think it could be good. Maybe not the best way to study drugs, but it could be good. Once again, to find the compound. But for example, in our case, that we define it that the mitochondria was the target we want to achieve. So it's not easy to build up to study the interactions in silico of these drugs, because mitochondria is an organelle, has a lot of functions in the biological system. So, for example, in this case, I think would not be the good one. The drug protein interaction, the in silico, I think it's a good starting point.
Thank you, sir. A question from Nitya, and it is the structural change in protein, example, amino acids change due to mutation, can affect the binding affinity of compounds?
Well, yes. So there is a field of computational biochemistry that is dedicated to this. For example, when you do I'm not the expert on this, but I have colleagues of mine that they are looking on this. For example, molecular docking. So in the molecular docking, we have published in the past some collaborations with the people from University of Porto. So they try to find molecules to monoamine oxidase B inhibition. So they have been performing studies on molecular docking, try to see how the molecules interact with the active center of this protein. It is a fact that the mutations depends when the mutation occurs in the protein. If there is a change in amino acid on the active center of this molecule, for obvious reasons, the molecule cannot fit anymore with the pocket of this enzyme. Okay, so I would say yes, but it depends where the mutation in the protein occurs. If the mutation in proteins occurs in the active center, in the pocket of the molecule of the enzyme, the molecule we have can be not fit anymore in the pocket. Okay, if it's outbound amino acid that is mutated, it may sometimes do not affect the activity of the compound. But for this in this case, I would say that molecular docking by computational studies are the most effective way to do that, because you can also modulate if you have the mutated protein crystallized, you can do molecular modeling and then do the affinity studies with the different compounds that we have to see if they lost or gained affinity. I think there is no more
Thank you,
questions
sir.
for now.
Thank you, sir, for answering all the questions asked by our participants.
You're
Here, the
welcome.
participant.
I have a question. Can I?
Yes, sure.
Thank you. Thank you, sir. That was a wonderful and detailed presentation. Thank you for that. I have two questions for you. One
Yep.
is that I just wanted to know about the drug. Does the drug target the mitochondria of the liver? Number one, only the mitochondria of the liver. And the second one, looking at a bigger picture, what are we actually asking the mitochondria to do? Are we asking it to burn the fat to produce ATP? A bigger picture, what are we expecting from the mitochondria point of view when we are targeting mitochondria?
Okay.
Thank
So, okay, thank you for your questions. So, first question, I would say no. This is not specific to liver mitochondria. As far as we know, this molecule reach every organ with mitochondria. Okay, so they accumulate within mitochondria. As long as they reach the organ, they can accumulate within mitochondria of all organs, which means similar molecules developed by different scientists in the field. They prove that molecules with these characteristics can accumulate in liver, muscle, heart, and brain, among others. So organs that have mitochondria, which is basically all of them, can be accumulate these molecules. So it depends the content of mitochondria. Then, so for example, cardiac heart has different content in mitochondria than liver, and brain has different mitochondria content than liver as well, and muscle, etc. So it depends on the content of mitochondria that the organ has. But in theory, these molecules accumulate in all organs that have mitochondria, active mitochondria. I did not present here, but that is something that we have some data, and we are keep accumulating data on this, is trying to prove that even if this molecule accumulate, for example, in heart and brain, they have no side effects on these organs. Okay, this is one of the main topics on my talk. It's the side effects, lack of efficacy and selectivity. Okay, we don't have too much selectivity for liver. We have selectivity for mitochondria, but not for organ selectivity. But we are now proving that there is no side effects, even they accumulate in heart and brain or muscle. There is no negative effects on this molecule in these organs. Okay. Second one, what we expect from this. So what we expect from mitochondria. Okay, so from we expect to improve mitochondrial function or decrease mitochondrial oxidative damage. Okay, so as I told you in the presentation, this slide about the Janus phase of mitochondria in non-alcoholic fatty liver disease, what we expect is that by constantly giving this compound to the mice, what we observe and what we believe is that we strengthen mitochondria. Okay, so by increasing by increasing antioxidant defenses, by increasing the number of mitochondria, the number of mitochondria per cell, and obviously in a later stress situation like the huge amount of fatty acids, we expect that mitochondria are more prepared to deal with the oxidative consequences, being fatty acids, being ROS production. So if the new pool, we believe that we have a new pool of mitochondria, and that this new pool of mitochondria are more resistant to oxidative damage. So in a global picture, we think this strategy can delay the progression of the disease. Okay, I'm not sure if this was the question, but in the big picture, we believe that this molecule can delay the progression of the disease by delaying the Janus phase, let's call that the Janus phase of mitochondria, the turn on of mitochondrial dysfunction. If mitochondria is more resistant to oxidative stress, it's less susceptible to damage, it's less susceptible to be in dysfunction, and the disease will be delayed. That's the big picture we expected.
Thank you so much, Sayed. Thank you. Yes.
You're welcome.
Thank you, sir.
You're welcome.
Is there any other questions from the participant side? If so, you can please post it in the chat box, or either unmute your mic and clarify your doubts.
Yeah. I think there is no more questions.
Thank you, sir, for answering all the questions asked by our participants. So here the participants are requested to fill the feedback form which is posted in the chat box, please.
Yeah, thank you. So I will stop
Thankfulness
my presentation. I will stop my presentation. Okay. And,
Thankfulness is the beginning of gratitude, and gratitude is the completion of thankfulness. Now I request our beloved Assistant Professor of Department of Zoology and convener of this conference, Dr. S. Manivandan sir, to deliver the oath of thanks.
Good evening all. Am I audible?
Yes,
Yes, sir,
sir.
you can continue.
Okay.
Yes, sir,
It
you
is
are
a great
audible. You are audible.
Okay. It is a great privilege for me to propose vote of thanks on this great occasion. The first owe my sincere gratitude our respected and most distinguished President Dr. M. Arichami sir for this blessing in each and everyday activity. I extend my heartfelt gratitude to our most esteemed Honorable Secretary and Director Dr. C. A. Vasiki Madam who encourage and motivate us in all our endeavors. It is a great time for me to thank our most respected Principal and Head Department of Zoology, Dr. M. Lakshmanasamy sir, for this encouragement and support. I express my sincere gratitude to our most respected former Vice Chancellor of Tiruvallur University, Vellore, and former Registrar in charge of Bharatiyar University, Coimbatore, and our Honorable Emeritus Professor, Dr. K. Murugan sir, for delivering them special address. We are all inspired by your great words, sir. I also take this opportunity to honor our most valued guest of this session, Dr. Jose Teixeira, research scientist, Center for Neuroscience and Cell Biology, and the Center for Innovative Biomedicine and Biotechnology, University of Coimbra, Portugal, for delivering a special lecture on toxicological and pharmacological effect of new molecules, who shared his knowledge with us, and the session was very inspiring. I also thank Dr. S. Palsamy sir, Dean Research and Development, Dr. Madan Sangan sir, Dean Academics, and Dr. S. Sivapragan sir, Associate Dean for this support. I must thank our head in charge, Dr. S. Binukumari, ma'am, who is with us all the time and encourage us. I also extend my thanks to all the faculty members and non-teaching members who are the pillars of the successful completion of this program. My gratitude to all the participants whose involvement made this session more valued. On behalf of Department of Zoology, Congrado Arts and Science College, and on my own, extend my heartfelt thanks to one and all present here and encourage to complete this session successfully. Thank you.
Thank you, sir, for expressing your gratitude through your honorable words.